Okay, so we're on our last unit here, unit 6, which is about the reproductive system. So we'll be talking about chapter 28, which is we're going to go over the reproductive system structures and then how the gametes are formed. And then chapter 29 will be covering pregnancy and development. So basically what happens if the gametes do fuse together and the development of the embryo and fetus, and then pregnancy stuff. So that's our last unit. So let's go ahead and begin with chapter 28. Chapter 28 is going to be talking about the reproductive system, as I mentioned. The reproductive system, its function is to ensure that species are going on, right? Hold on, let me check. Okay, just checking the chat box here for just a moment. All right, so chapter 28, or a reproductive system, the function of the reproductive system is to make sure that species continues on, and so it's reproduction. That's what that means. The main organs of the reproductive system are called gonads. Gonads are going to produce your gametes and your sex hormones. The gonads in males are the testes. The gonads in females are the ovaries. And then the gametes in males are sperm. Gametes in females are egg cells or ova. And the sex hormones are also going to be different in males and females. So for males, the main sex hormone is testosterone. For females, the main sex hormone is or are progesterone and estrogen. We'll then also talk about accessory reproductive organs. And so in males, this is going to be a set of tubules, different tubules and such, as well as various glands, and then some external structures that are going to aid with reproduction. With females, we'll also be talking about some accessory structures. So these are going to include fallopian tubes and the uterus, vagina, and then some external structures as well, which we will see actually in a moment. The gonads don't become fully functional until the puberty. So they are there, they're present, but they're not actually functioning or doing anything. When we look at the female reproductive system, we'll see that actually some stuff is going to start happening before the female is even born in her gonads. But the full functionality of gonads, even for female, begins in the puberty, okay? Typically, females are going to produce and release 1 oocyte a month. For males, this is about 100 million sperm a day. So they produce about 100 million sperm a day. Quite a difference there, okay? 100 million per day versus 1-ish a month. And I'm saying ish because some females sometimes do release more than 1 oocyte, but typically it's once a month, okay? So that's that. So let's look at some homologous structures here. And this is what this table is showing us. Just so you understand what homologous structures mean, homologous structures are corresponding structures. So they, what we mean by that is that they originate from the same primordial tissue during the fetal development, and then they develop into different structures. Like they're going to look different, they're going to have slightly different function, but general function is going to be the same. So a first homologous pair here in this table is ovaries and testes. These are going to be structures that produce gametes and sex hormones, right? So the general function is the same. Now, when you look at specifics as far as what the gametes are and what the sex hormones are, there's a difference, right? But the general function is the same, and they also both originate from the same primordial tissue. And that will make a little more sense when we actually talk about the genetic and phenotypic sex at the end of this chapter. I almost feel like they should actually include that here. Actually, let me see if I can find that image. Okay, where is it? So like right here, okay? When we look at this, we'll be talking about the development of the reproductive structures during embryonic and fetal development. So actually, starting here. So here we're looking at an embryo that's about somewhere between weeks 5 and 6. And so you can see that the blue area right there, so right here, okay, that's a genital ridge. And this genital ridge will become either testes or ovaries, depending on the presence of hormones that we have there, okay? But early on, it's the same thing, right? It will just develop into a different structure depending on our chromosomes present and then hormones being produced. So that's what that means, developing from the same primordial structures, okay? Okay, and some other homologs here: clitoris and glans of penis, as well as labia minora and body of penis. Those two pairs both kind of have the same or similar function, and that's that they contain erectile tissue that's stimulated during sexual arousal and orgasm. Then labia, some protective structures include labia majora in females and scrotum in males. These are going to protect and cover some reproductive structures. And then a couple of glands here, the greater vestibular gland and bulbourethral gland. These both secrete mucin, and this is going to aid in lubrication in males. The bulbourethral gland is also going to serve sort of as a protectant for the urethra. So that's our homologs. Okay, now we're going to look at this area right here, or this image right here. And this is just showing you these two triangles, these regions here. We can talk about the urogenital triangle and then the anal triangle. And you can see one notable difference here between the males and the females is that the male, the angle is not as wide as in the females. And so you talked about this in AMP1 when you talked about the skeleton and the pelvis differences between males and females, right? So you know that in females, the pelvis, this triangle is going to be wider, and that is because it's so that it can accommodate passage of fetus eventually. And so there are these points that are going to mark these triangles here. So we have the urogenital triangle is going to be the pubic symphysis, and then the ischial tuberosities, and the anal triangle has those ischial tuberosities, and the coccyx is what marks that. So make sure that you are able to label or tell me which one is urogenital, which one is anal triangle here. That should not be super difficult. So that's just a little bit of intro here. Now we're going to look at a little bit of heredity. Now we're going to have heredity in chapter 29. So we'll be talking about like Punnett squares and how things are inherited later. But before we talk about gametogenesis here, we have to cover the basics as far as like how many chromosomes we have and just the basic language of genetics before we can talk about formation of sperm and eggs. So humans have 23 pairs of chromosomes, and notice I said 23 pairs of chromosomes, right? Different organisms have different number of chromosomes, okay? Humans happen to have 23 pairs of chromosomes. 22 of these pairs are called autosomes. So these are your chromosomes 1 through 22. We don't name chromosomes, we number them. So each chromosome is assigned a number, and they're actually numbered based on their size, okay? And again, we'll see a karyotype in chapter 29. And so you'll see all those chromosomes lined up. And so when you're talking about chromosome 5, you know, it's a chromosome that carries the same genes, you know, among all the people, right? So that's how we do the chromosomes. So 22 pairs are autosomes, and then one pair, the 23rd pair, is going to be the sex pair. So this is a pair of sex chromosomes. And for females, generally this is X and X, and for males, this is X and Y. There can be some variation here. We might mention some later on, but typically it's X and 2 X's for female and X and Y for a male, okay? So we don't actually, so we can talk about homologous chromosomes, The autosomes are considered homologous chromosomes. Again, homologous means that they're corresponding, and they, just like the homologous structures were corresponding structures where they had, you know, they were not identical, but they had kind of the same function. Our autosomes are corresponding chromosomes. So the autosomal pair, the 22 pairs of autosomes, are homologous chromosomes. So we said they're matching or corresponding. What this means is that they carry the same genes, but not necessarily the same versions of each gene, okay? This is because you have, the reason why you have 23 pairs of chromosomes is that you get 23 single chromosomes from your mom, and you get 23 single chromosomes from your dad. Then they come together and create that first diploid cell there. That's the first cell of the new organism, okay? So that's kind of how that works. So when I said that they have the same genes but not necessarily the same version, what I mean by that is, let's say that eye color was coded by one gene, okay? It's not, it's actually coded by like 5 genes, but let's just simplify it and say eye color, okay? And let's say that it's on chromosome 5. I'm totally making this up. I don't know what chromosome that is on, but let's just say that eye color was on chromosome 5. Well, the chromosome 5 from your dad could carry eye color brown, and chromosome 5 from your mom is still going to have gene for eye color, but it could be blue, okay? So all the chromosomes have the same genes, but not exactly the same color or feature or characteristic for that gene, okay? So I hope that that kind of makes sense there, okay? All the cells in your body that are not sex cells are known as somatic cells, okay? These cells have 23 pairs of chromosomes. We call them diploid cells, and we say that the number of chromosomes that they carry is 2n, where n is the number of unpaired chromosomes, okay? Then you have cells that are not somatic cells. These are the gametes, and that's the sperm and egg cells, okay? These cells are going to carry 23 single chromosomes, okay? And these are called haploid, and we designate them by letter N, okay? So only gametes have 23 single chromosomes. They don't have pairs of chromosomes. And then all your somatic cells, all the other cells in your body, are going to have 23 pairs. It is important that your gametes are haploid. It is important that gametes only carry one set of the chromosomes, because what gametes ultimately do is they fuse together to create the new organism, right? And so if you didn't have only 23 single chromosomes, but you had a diploid gamete and a diploid gamete, if they fuse together, you would have not pairs, but you would have 4 of each chromosomes. And having extra chromosomes in humans is not good, okay? It doesn't make us extra strong or, you know, superhuman. Even one extra chromosome can have severe consequences in our genetics. So we have to have gametes that are haploid. There are some organisms that can be polyploid, meaning that they can have 8 or 16 of each chromosome, but this happens in plants, okay, not in humans. Humans, no polyploidy, okay. We want to be diploid for our somatic cells and then haploid for the gametes, so that when they fuse together, they create another diploid organism, okay. So make sure that you know diploid versus haploid. Diploid, the di prefix should tell you that it's 2, okay, so that's pairs of chromosomes. And haploid almost sounds like half of the diploid, so that's just a single, 23 single chromosomes there. So this leads us into talking about meiosis, and meiosis is known as lessening. It's also known as the reduction division, and this is the cell division. It's a special type of cell division that is used when our body is producing gametes, okay? lessening is going, or meiosis is going to begin with a diploid cell, and then it will result in 4 non-identical haploid cells. Now, if you take mitosis, okay, you've already talked about mitosis in AMP1, I believe. The regular cell division, you start with a diploid cell, and then that one cell is divided into 2 daughter cells that are identical, okay? That's mitosis. Meiosis, you start with a diploid cell, and at the end of the process of meiosis, you'll end up with 4 non-identical haploid cells, okay? So that's the main difference between the two. Make sure that you write that down somewhere, and I'll actually write it down eventually at some point after we go through, you know, I show you the stages or phases of meiosis. Okay. So when a cell, we said that it starts with a diploid cell, right? So when a cell prepares for division, any cell, even if it's going through mitosis, there are certain things that that cell does during interphase. And part of that is preparing for division. So a cell that is going to go into gametogenesis or into meiosis is going to do these things as well. It's going to have to prepare for this division. One of the things that happens when a cell prepares for division is that it has to copy its DNA, right? So, you know, even for mitosis, that cell, when it's preparing for division, it has to create a copy of all the DNA so that the 2 daughter cells each have their own full complete copy, right? So before meiosis, DNA is replicated as well. For this this image right here, I want to kind of talk about our chromosomes and what we're going to see. So you begin, and we're just looking at one chromosome, right? So right here at the bottom, you're looking at a single chromosome, okay? In the middle here, you're looking at a pair of homologous chromosomes. Okay, these are the same chromosome as far as number and the gene, but they're not identical, they're homologous because one is from mom and one is from dad. Okay, so when replication of DNA happens, and you would have 23 pairs in the regular cell that hasn't replicated its DNA yet, you would have 23 of these, 23 pairs like this, okay, homologous pairs. All right, so what happens during replication of DNA is that each of these chromosomes is going to replicate itself, or is going to be replicated, okay, by the cell. So this red one replicates, is replicated, and then this one is replicated. So before the cell goes into the division, it will end up with 23 pairs of replicated chromosomes like this. 23 pairs of replicated chromosomes. So these two, hold on, how do I best show this? So this red one, the red X-like structure, and the blue one together, these are homologous chromosomes. Homologous replicated chromosomes. And these are non-identical. Okay, I'm not identical. That says identical. I'm kind of running out of space there. Okay, so the red and blue, they're replicated homologous chromosomes. The red and blue are not identical with each other. Okay. But when you look at this and this, the 2 red structures, so like this one and this one, these are called sister chromatids. And they are identical to each other. Okay, so the 2 red are sister chromatids. They're identical to each other. Same goes for the two blue parts, okay? The sister chromatids are identical because they're a result of a copy of the DNA, okay? This is when the cell made another copy of the DNA. So the two reds and the two blues are identical, but the red and blue are not identical, okay? So that's kind of how that goes. After the replication happens, the sister chromatids are held together by a structure known as the centromere. The centromere has this region on it that's known as the kinetochore, and that's what the spindle fiber will attach to eventually whenever we're moving chromosomes to different sides. Okay, so that's kind of how this works. All right. So again, sister chromatids are not the same as homologous pairs. Okay. Now, let me see. Let's try this. Can you see the questions? I don't know, it doesn't really show up for me. I just see my shared screen. Okay. Okay, so if you're still answering, that's fine. But the two questions that I have here were testes and ovaries are homologous structures. Is that true or false? And then the second one is labia minora and scrotum are homologous structures, true or false? So let me see how many people are here today. 11. Okay, the first one was pretty easy, right? Testes and ovaries are homologous structures. That is correct. These are the gonads. So, you know, rather easy. Now, the second one, I kind of did a trick there on you a little bit. So labia minora and scrotum, they have, they're actually not homologous structures. So if you think back to that table that we saw, labia minora is homologous with the body of penis, but the scrotum would be homologous with labia majora. Okay, so they're similar terms, but these two are not homologous. Labia minora has that, contains that erectile tissue. Scrotum is a protective structure of the gonads. So that's that. So I'm still, this was the first time I actually did a poll like this. So, all right. And I think I have another one that I wanted to do. Okay. How about this? Oh, launching the poll would be nice, right? I'm just like staring at you. Okay, so how about these two? Okay, so that first question, humans have 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. Okay, so out of those 23, 22 are autosomes, 1 is sex chromosomes. And that statement, everybody that has answered agrees that that statement is true, so that's correct. And then second question says cells containing 23 pairs of chromosomes are called haploid, and cells with 23 single chromosomes are diploid. And all of those that have answered have agreed that this is indeed false, okay? It's the other way around, right? So 23 pairs, that's diploid, and then 23 singles are haploid, okay? All right, so I'm gonna end that. And that's that. And then, let me see, oops. What else I have? Okay, what does each cell have to do before it can divide? Oh, I keep forgetting to launch the poll. Okay, some people answer very quick. Good job. All right, 8 people. Good. Oh, that's fine. No, I just forgot to launch the poll again, so I'm just, waiting for the answers, and I, didn't start it. Okay, yeah, that's fine. All right, so what must happen before every cell is able to divide? It has to replicate its DNA. That's what I was going for here. And most of you got that. Chromosomes have to condense. Chromosomes actually don't condense until prophase 1, so it wouldn't be part of that interphase. Maybe I'll word this differently next time, so that like I'm talking about, like before it enters the division. Actually, when the DNA is copied, in order for our cells to be able to copy the DNA has to unwind. It has to be loosened up so that it can be read and copied. And then the chromosomes will condense when we enter the cell division. The other two, I don't know, I just wanted to have some other answer choices there, but they're like, yeah, they're not really relevant to them. Okay, so I'm gonna end this poll, and that's that. Okay, so let's go ahead and look at the events of the meiosis. So what happens when we're actually going through the division here? Meiosis is, because it's a reduction division or a lessening, and we're trying to get to 4 cells eventually, it is going to involve 2 divisions, okay? So the division is going to occur twice in that one meiosis process. So we have to divide it into meiosis 1 and meiosis 2. The phases are the same as in mitosis. However, you have to be specific and with whether we're talking about meiosis 1 or meiosis 2. So the order is the same as with mitosis, the PMAT order: prophase, metaphase, anaphase, telophase. You're going to see some similarities between mitosis, but you have to specify, are we talking about prophase 1 or prophase 2? Because there are slightly different events happen there, okay? So let's look at the prophase 1 here. It's showing you just 3 chromosomes here because 23, that would be a lot to follow. So we're just looking at 3. But keep in mind that this, a human cell would have 23 pairs that are doing these things, okay? So we're just looking at 3. So in the prophase 1, remember, cell enters prophase 1 after it replicated its DNA, and that's why you see the replicated chromosomes here. What happens here is the nuclear envelope disappears. Your chromosomes are going to condense. Now the condensed shape is going to help eventually splitting the chromosomes apart. So the chromosomes condense, they form those condensed structures there, and they become visible because nuclear envelope disappeared, right? The homologous chromosomes actually are going to find each other during prophase 1. So in mitosis, chromosome condense, nuclear envelope disappears, it's the same, but the homologous chromosomes during mitosis, during regular cell division, they don't care about each other, okay? They're not gonna find its homologous pair. During meiosis, during prophase 1, homologous pairs actually do find each other. They come near each other. And so what happens is you see this formation of what we call a tetrad, and that's this right here. That's one tetrad, and then you have second and third. Okay, so you have 3 tetrads. In humans with the 23 chromosomes, you would have 23 tetrads altogether, okay? But these homologous pairs, homologous replicated pairs, when they come together like this, they form a tetrad. Tetra prefix means 4, okay? So when you look at each tetrad, if you were to count single chromosomes that you have in there, you would see 4, okay? There is, and I'm gonna draw on these, okay? There is one here, oops, one here, and one here, okay? So that's 4 single chromosomes. All right. Yeah, because each replicated chromosome has 2, right? 2 chromosomes. And so you would have a total of 4 chromosomes. At the end, what you're going to end up with, remember from meiosis is going to result in 4 non-identical cells. One of each of those chromosomes from each tetrad will be in a different separate cell, okay? So tetrads form. When tetrads form, this means that these homologous chromosomes are near each other. Now remember that homologous chromosomes have same genes on them. And so during this time when they are near each other, some chromosomes exchange parts of each other with each other, okay? So they exchange their parts with each other, and this is known as crossing over. So a piece of the father's chromosome can switch with a piece of the mother's chromosome and vice versa. Here, what it shows you is just ends kind of swapping. The crossing over is random. It could be middle sections switching with each other. And so it's every time crossing over happens, it happens differently. And this helps with genetic variation amongst populations. Okay, so it's basically, it's literally like mixing genes there at that level, okay? So while your body is producing gametes, your mom's chromosomes, your mom's genes and your dad's genes are mixing, okay, with each other. So that's what's happening there. Crossing over is only happening in prophase 1. It does not happen in any other phase, okay? Only in prophase 1. Yeah, so that's that. That's prophase 1. That's the important part about prophase 1. Crossing over happens, tetrads form, and that's that. The second phase is going to be metaphase. Metaphase, I want you to think meta for middle. So metaphase means that these homologous pairs are going to line up along the middle of the cell. Now, in mitosis, you would see just chromosomes lining up in the middle of the cell. Here in meiosis, we have the pairs line up along the middle of the cell. So it means you have 2 rows of chromosomes in the middle. Meiosis would just have 1. Okay, but here you have 2 rows of these homologous chromosomes, and they're kind of hanging out there in the middle. Then anaphase comes. Anaphase means like we're separating these chromosomes, but we're not separating the sister chromatids. So we're just separating the homologous pairs. So one of the homologous pairs goes to one side and the other one goes to the other side. So we're splitting the homologous pairs, not the sister chromatids yet. We're just splitting the homologous pairs. And then telophase 1 and cytokinesis. Telophase 1, you are going to see the nuclear envelope briefly reappear. Chromosomes kind of could relax a little bit, but not for long because we're going to go to the next phase right away. Cytokinesis will finish the division of the cytoplasm here. Okay, so now when you're looking at these 2 cells, this one and this one, and you look at the chromosomes, you have the replicated chromosomes, but do you have homologous pairs at this point? Do you see homologous pairs in those that I just highlighted? Yes, no, okay, fine, I'll answer it myself. So the answer is actually no, you don't see the homologous pairs anymore. You see replicated chromosomes, but they're non-identical. When you look at the top cell and the bottom cell, you can see that the smallest chromosome, that's different. The bigger ones look like they might be the same. But again, we don't know what the genes have shuffled with what, you know. So these went through crossing over, and you split the homologous pairs in anaphase. They were separated. Homologous pairs were separated in anaphase 1. So your resulting cells after meiosis 1 are going to be already considered haploid cells because you don't have homologous pairs. You have replicated chromosomes, okay? And they're non-identical. Okay, so here after meiosis 1, you get 2 non-identical haploid cells. Okay, so meiosis 1 gives you 2 non-identical haploid cells. We already considered them haploid, all right? Now, remember we said that before every division, cell replicates its DNA? Before every division, except for division of meiosis 2. So right after meiosis 1, we're going to go directly into meiosis 2, okay? We're not replicating that DNA anymore. So meiosis 2, we're going to have prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis. And what happens here is very similar to mitosis, except instead of having 46 of these replicated chromosomes, now you have only 23 and 23 in each cell, okay? So here we don't have the homologous pairs. We just have those replicated chromosomes. They appear, they condense, nuclear envelope disappears, so they become visible in prophase 2. There's no crossing over here, okay? It's just regular prophase here at this point. Metaphase 2 is going to be chromosomes lining up along the middle of the cell. Now you just have a single plane. Okay, because we don't have the homologous pairs here anymore. So single row of chromosomes or single plane of chromosomes, if you're thinking in a 3D fashion. Okay, cell is kind of, it's not flat, but more like a roundish cell. And so they're going to be along that middle plane of the cell. Okay. The anaphase will cause the replicated chromosomes to split. So it's pulling apart sister chromatids now. Okay, in anaphase 1, we're splitting homologous pairs. In anaphase 2, we're splitting the sister chromatids. All right. And ultimately, when they're pulled to the opposite sides of the cells, we end up with 1, 2, 3, and 4 haploid non-identical cells. Okay. And these are going to be your gametes. If this is spermatogenesis, if this is following the male sperm development, each of these cells will become sperm. Okay, so one diploid cell in the beginning produces 4 sperm that can go and fertilize an egg. Okay, and we'll see this when we look at the female reproductive system. In females, only one of these 4 will actually be the oocyte. The other 3 will become what we call polar bodies, and they will degenerate in the woman's body. Okay. So, but we'll look at that later when we actually look at the female reproductive system. So, meiosis does take a little bit of time to go through. I want you to really think about the differences between mitosis and meiosis. How do I, hold on. How do I get a whiteboard here? There must be a way, because I wanted to write down the summary of the differences between these. So, let me see. No, that's not what I wanted. Okay, like this, I guess that will work. Okay, so for meiosis, you're going to end up with 4 non-identical haploid cells. And mitosis gives you, everything will be different about mitosis, except for the word cells. Okay, so you don't get, in mitosis, you don't get 4, but you get 2. And you don't get non-identical in mitosis, you get identical. And then it's diploid cells. All right. This is an important concept from chapter 28. So make sure that you can tell me, you know, that meiosis gives you 4 non-identical haploid cells, and mitosis, what did I say? Oh my goodness. Okay, meiosis gives you 4 non-identical haploid cells. Mitosis gives you 2 identical diploid cells, okay? And they do sound similar. So sometimes, you know, you might want to mix them up. So make sure that you really slowly think about it and get to the correct answer there. So that concludes that that part. I'm going to actually make a female reproductive and then a male reproductive video separately, because it is already 45 minutes. And if I were to jump into the female reproductive system, it's longer than male. So that would take quite a bit of your time right now. So I'll post those as the recordings, and this will be it for the live part. Make sure that you watch those. You know, I make them so that, you know, you can learn from them, hopefully. And don't binge watch this stuff, okay? It doesn't quite work very well if you're trying to binge watch this the day of the exam. So, you know, make sure that you actually take some time to sit down with it, you know, have pen and paper, make notes within your notes that you have taken already from the textbook, and use those videos kind of like that. If you watch them earlier, you know, that gives you opportunity to ask questions too. So binge watching AMP is not very good. Okay. So, wait, I want to stop sharing. Oh, I stopped sharing. I want to stop recording.